There are three types of RNA that are involved in transcription and translation. One of them you've already heard, messenger RNA. The other two are ribosomal RNA, or rRNA, and transfer RNA, which is tRNA. Each one of these is a type of RNA that has specific function in the process that we are talking about. And by process, I mean transcription and translation, because you don't really have one without the other. I can't make a protein without having notes to make it, and I don't take notes for nothing. I don't go to my grandmother's house to get the chocolate chip cookie recipe and then never make chocolate chip cookies. I have both of them interconnected, and they have three different types of RNA that function in various aspects of this beginning to end process. So mRNA is the one we were just talking about. mRNA, aka, or also known as messenger RNA, is my intermediate between my DNA and my protein. These are the notes that I take from the DNA instruction manual to make the protein that I want. Each three-letter sequence of nucleotides is a code for something specific. So we call each three-letter sequence a codon. 3 nucleotides equals a codon. Each codon is a code for start, stop, or a specific amino acid. Now remember, an amino acid is a building block for a protein. An amino acid is equivalent to 1 Lego. The protein is equivalent to building something with Legos. I might build the Death Star, like I said before. I might build a castle. I might build, I don't know, a car. If you've seen the Lego movie, I might build a spaceship. Either way, the specific amino acid placement and sequence gives us the ability to build something unique. I may have, because we just talked about it. We talked about DNA replication, and we talked about DNA polymerase, we talked about helicase, we talked about RNA polymerase, we talked about topoisomerase. All of those are proteins, and each one of those proteins has a recipe in the genetics. What the sequence of amino acids are to build those varies. That's why they are different from each other. I can have peanut butter cookies. I can have monster cookies. I can have oatmeal chocolate chip cookies. I can have red, white, and blue cookies. I can have sugar cookies. I can have chocolate chip cookies. I can have double chocolate chip cookies. All of these are still cookies, but they're different from each other based on the recipe. All of the things that I just mentioned are proteins, but they're different from each other based on the recipe, based on the sequence of amino acids that I have. So that's mRNA. This is how we get the recipe out of our nucleus so that we can get it read. Don't freak out. I'm not asking you to memorize this. But I do want to show you something. Remember, I said every 3 letters codes for a specific amino acid. 3 letters. So it's a 3-letter sequence, right? So there's position number 1, position number 2, and position number 3. This is the first base in the sequence. So this is number 1, looking here. This is number 1. Here, going across this way, is number 2 in the sequence. Here is number 3. Again, going down the sequence. So, I'm just going to randomly pick. Let's say UGA. What does that code for? My first letter is U, so it has to be here somewhere. My second letter is G. So this is U and G, right? My third letter is A. There's A. It's a stop. So what does it code for? Remember I said that when we go, there's a sequence that means stop. There's a stop sequence. This table tells you exactly what each three-letter code means. So instead, let's go with something different. Let's go with C, A, G. Why not? First letter C, so that means we go this way. Second letter is A, so that's going to be here somewhere. Third is G, so that's this one. CAG. This is the amino acid glutamine. Every three letters means something specific. Now, I do want you to notice something about this as well, though. You'll notice with the glutamine, I actually do have two sequences right here that both code for glutamine. And if you look here, there are 4 sequences here and 2 here that all code for arginine. There's redundancy built into the system. And if you notice here especially, the first 2 letters are the same for all of these variants of arginine. Now, why would that be important? What's the dealio there? If I have a mutation that changes that third letter, there is a reduced chance that I will get some weird protein in that spot instead of arginine. It would be like me putting salt, 2 cups of salt into a cookie recipe instead of sugar. They don't taste the same, right? So when those cookies were finished, you probably wouldn't like them, right? If, however, I have alternatives like I can use raw sugar, I can use white sugar, I can use brown sugar. Technically, they're all the same. They're all sugar. Yes, it might slightly change the recipe, but the recipe will still work. I have one recipe that called for half a cup of white sugar and half a cup of brown sugar. And because I wanted the cookies to look a specific way, I took the brown sugar out and just replaced it with white sugar. People love those cookies. Does it still work as a cookie? Yes. If I change that third nitrogenous base, does it still work to be arginine? Yes. Having the ability to make small changes but still get the same results basically means that, or hopefully helps in the fact that some small mutations may not change anything. It may not be detrimental to our protein that we need. Now, are there mutations that definitely mess everything up? Yes, of course there are. There are always going to be mutations that can mess everything up. But having redundancy in the system ultimately means if the third base in the sequence is changed, we still get the same result. Can a mutation be detrimental? Definitely. If you change the first letter in the sequence, you may still get arginine, or you may get serine. You may get a stop. You may get cysteine. You may get tryptophan. You could get glycine. And each of these is a different amino acid, which leads to a different protein. It would be equivalent to putting glass on the rims of your tires in some cases. If I put glass from my windshield on the rim of my tire, that's not going to function correctly. If I use salt instead of sugar in a cookie recipe, that wouldn't function correctly. If I changed what kind of sugar I was using, though, probably it would work the same exact way. I would still get an edible cookie regardless of whether I used sugar in the raw or brown sugar or white sugar. Taste might slightly vary, but the cookie itself would still taste sweet and be edible. With redundancy like this, hopefully we still get the same results. But the possibility does exist that we won't if the mutation is bad enough. Ribosomal RNA. Our RNA, ribosomal RNA, is a girder in a building. It's actually part of the building material that makes our ribosomes. Remember, ribosomes are the hamburger bun that reads the messenger RNA. Ribosomes are the protein factory where we take our messenger RNA, our notes, and we make it into a sequence of amino acids. This is the reader. But as far as what part ribosomal RNA plays in that, it's used to build this. If you look at the figure, anything that's gray and anything that is blue is the ribosomal RNA. It's used to build this structure. The last type of RNA we're going to talk about are transfer RNA. Transfer RNA do exactly what they say, what the name suggests. They transfer something. Transfer RNA has two primary parts that are important. They have an anticodon. Remember, the codons are the codes for the amino acids. They have the complement to that called an anticodon that sits on this side of our transfer RNA. The other thing that they carry, almost like somebody carrying a backpack with something specific in it, is the amino acid that matches with the codon on our messenger RNA. As the ribosome reads the messenger RNA, it sees a 3-letter sequence and says, I need this. Basically, this is what the sequence is telling me. In the case of isoleucine, there are 3 codes in the mRNA that are possible to give me isoleucine. AUU, I'm going to go back to here. If in the sequence of mRNA I have AUU, AUC, or AUA, that indicates I need isoleucine. Good so far, I hope. I'm going to come up with, well, I've already written it down, but just a generic code, all right? A generic, this is the code kind of thing. AUG, UCC, UAU, AUU, and UGA. AUG is start. That is normally our start codon. Are there exceptions? There's one. But I'd say probably 75% of the time, your start codon will be AUG. UGA is stop. Remember I said we had 3 stops? UGA is stop. But these sequences here are all a specific amino acid. UCC is for serine. UAU is for tyrosine. And AUU is for isoleucine. Isoleucine. So I have a tRNA for isoleucine. The thing is, going back, I have 3 different codes that are for isoleucine, correct? Correct. That means that I need to have 3 different tRNAs, one for each code that specifically carry isoleucine. So if you look at these 3 tRNAs, every single one of them carries isoleucine. That's the amino acid that's sitting on the opposite end. The anticodon is the complement to the codon. Now, why do they call it the anticodon? Because it's not the exact same code as what you find in the mRNA. It has to be its matching partner. So for AAU, which is one of the codes for isoleucine, I would need complement. Across from A goes U, across from U goes A. I would need a UAA to complement that. The anticodon portion of my tRNA has the complement. So this would be UAA. When my ribosome is reading and gets to AUU, this is the specific transfer RNA that brings me isoleucine, because its anticodon matches the codon. It's the complement. Remember, isoleucine has 3 codes. So if I were to repeat this sequence 3 times, each position here, even though the sequence is different, still codes for the same amino acid, isoleucine. If you look at this table, you'll see AUU, AUC, AUA. All of them are for isoleucine. But if I need something that is the complement, this will not work for AUC. This will not work for AUA either, as an anticodon to complement the sequence in sequence number 1. Do I still need isoleucine in both of these? Yes, but I need a different transfer RNA that still carries isoleucine. So in the case of number 2, sequence number 2, I would have a transfer RNA that has the anticodon, the complement to AUC. So I would start here, put a serine, put a tyrosine. When I got to this, it would be this transfer RNA that gives me the complement sequence to bring the right amino acid. In the case of sequence 3, is AUA still isoleucine? Yes, but neither one of these anticodons matches AUA. So I would need another transfer RNA or tRNA that has the anticodon UAU that would complement the AUA. Every time I run into, I'll say, the AUC sequence in a messenger RNA, it's this tRNA with this specific anticodon that brings me isoleucine. The kind of cool thing about transfer RNA is once I drop off that isoleucine, I pick up another isoleucine, and I wait for my turn to come again. I can use this transfer RNA over and over and over, just reload it and reuse it. And whenever the AUC sequence comes up, whether it's in this or in another protein, he drops off isoleucine. If I have a huge, long, 1,000 length, amino acid code, and it happens to have an AUU in the sequence, it's this guy who has the anticodon for AUU that will bring me that isoleucine. If I have another one, another messenger RNA that has a codon that is AUA, it's this third option that will bring me the isoleucine. In all of those cases, do I get an isoleucine? Yes. The only difference is who delivers it.